to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 48th episode of the Weekly Bioanalysis, the official podcast of KCAS, Bioanalytical and Biomarker services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I've been in the bioanalytical LCMS space for over 26 years. I'm here with my co-host Dominic Guarino.
Hello, my name is Dominic Guarino and I've been in the ligand binding assay biomarker cell gene therapy space for a little over 27 years.
Dominic and I are senior scientific advisors at KCS, and either or both of us are available to answer any questions you may have regarding the podcast or any of KCS's services. I'm located in upstate New York, Dominic is in Kansas, and our producers are in Missouri and Kansas City, so we're all over the map. We're thrilled to have you listening to the 48th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you're joining us for the first time or if you're a regular listener. As many of you know, if you've listened to any of our previous episodes, we started the podcast when COVID restrictions prevented us from continuing business as usual. We were unable to meet colleagues face-to-face, but we want to continue communicating with all of you. The podcast was an idea we'd always had, but it suddenly became the perfect medium for us to employ. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. Then we'll wrap up and give you a little teaser for the next episode, letting you know what is coming in the future. So again, we're fired up to have you here, and we're looking forward to a fun episode today. To kick us off for podcast number 48, Dom is going to go over today's podcast topic a bit before we jump into the news and resources section. Dom.
Yeah, John, as always, we will do the news segment, the news and resources segment, and that it will have our non-COVID and COVID issues. And then today's main topic is a very exciting topic near and dear to all our hearts, and that is ADCs, John, and we'll go over Antibody-Drug Conjugates, and we'll tackle it from many different angles. Not only the power of them and the combination of the therapeutics that are putting together to form ADCs, as well as some of the bioanalytical challenges, and as well as what are the approvals and what it's treating. But definitely good stuff there, John. And the ADC, the antibody-drug conjugate space, is saving lives, John. So with that, and then of course we'll probably wrap up with some feedback and then talk about our topic of choice for next week if time permits. But John, with that, let's just jump right into the news.
Dominic, do you want to start on this one?
Yeah, John. So the first news item, of course, we're going to talk about something COVID related. It's ivermectin. I don't know if I said that right. I'm going to go with ivermectin or ivermectin.
I'd use ivermectin.
Yeah, ivermectin. So this, John, this is ivermectin has made a splash is the term that comes to mind, and I think in a very bad way, right? So ivermectin is a medicine approved back in the late 1970s for parasitic conditions such as river blindness, intestinal worms, and head lice. It's also prescribed by veterinarians for other such infections in livestock. So back in like April of 2020, some Australian scientists reported it could help reduce could reduce some of SARS-CoV-2 viruses. So ivermectin suddenly has now made a splash through an Argentinian study, and then even into here in the States with a certain podcaster / Hollywood type, I'm not going to say his name, really kind of perpetuating this. I'll call it lie, John. I think that's the word that comes to mind.
Yes.
This is a lie, a straight up lie. So, John, you seem to have a little more. I'm going to let you go a little bit more in detail, and then I'll share more about my thoughts on this. But this ivermectin is the epitome of things that go wrong when science goes bad, right? When people say, "Oh, listen to the science," this is the opposite of that, right, John? Like people should not listen to this gobbledygook and rhetoric around this particular small molecule.
Yeah, so we have about 4 different articles on this, all coming at it from different ways, but you're right. The background is it all started with a study in Australia where they're looking at actually in vitro whether it whether ivermectin could be effective against SARS-CoV-2. But the key takeaway from that study was that the doses were so high that actually there was extremely high concentration. If it was dosed at equivalent level to into humans, you couldn't actually get it into the human body, and so it was much, much, much higher than what could be achieved in the human body.
And John, let's take a pause there, right? So the
Yes.
first study is flawed there. So people are like, "Oh, this paper says it's working well," but the reality is, you couldn't pump enough of this drug into somebody at the levels they were seeing it. So, John, in those types of settings, you could use. I'm sure I could use vinegar or something like that to show it could kill something, right? So, sorry, I want to set the
That's fine.
stage around the audience to
Absolutely.
know that, like,
Absolutely.
this lab over here is basically saying this isn't working. That's what they
Yeah.
concluded in that
Yeah.
study.
But so that's the starting point. There's been additional studies that people will cite, as they showed really good data that talked about effectiveness for ivermectin. And actually, there was a physician talked in front of a Senate committee in December, described it as a wonder drug, and actually talked about a trial from Argentina. And I wanted to dig into some of the background, because we'd seen the headlines about people taking ivermectin, refusing other medications, and talk about ivermectin as a wonder drug, and livestock uh, sellers selling out of ivermectin because people were taking the livestock intended drug and administering it to themselves. That was then having an effect on emergency rooms because people were having side effects, etc. So I wanted to really try and figure out, well, where did this come from? So the trial that was cited from Argentina, which sounds great, is when you actually dig into it. And there is this, I have to cite, I think this was BuzzFeed did a really good sort of step by step walk through what was flawed about this paper. It's not the only one that, and actually in parallel, there was another study in Egypt, which was a large study. And because of the size of the Egyptian study, the conclusions from that really weighted the overall ivermectin as a wonder drug. If there was other studies that said it didn't work, this Egyptian study was so large saying it worked that it actually biased the overall conclusion of all these different studies. Now, the Egyptian study, I would step into that first and then come back to the Argentinian one. It was actually a student in London, as part of his masters, was assigned. Um, the guy's name was Jack Lawrence. Was actually assigned just a paper to dig into it and say, well, what do you talk about the research that was done in this paper? And he was assigned a preprint about this Egyptian study. And once he started looking at the data, he started noticing serious flaws. Just starting with the introduction, which has basically been plagiarized from all over the place and run through translation software, such that some of the terminology just became gibberish because the translation software didn't really understand what it was talking about. Started digging through, didn't make sense in terms of when they said the study was carried out, when people recruited. So it said it started on the 8th of June, but some people who were listed as recruits actually died before 8th of June. He also shared data with some Australian researchers who, when they ran through it, they said we could see pieces of data that were cloned from one patient to another, and then subtle details were changed to make it look like the data was different, but in actual fact it was the same data. The Egyptian paper was eventually, as a result of the work that Jack Lawrence and these Australian researchers done, was pulled as a preprint, so it's actually not in the literature anymore. So we'll then go back to the Argentinian one. Sadly, that also has a lot of inconsistencies that basically says either the study didn't happen or the study happened in a tiny fashion and they inflated it. There's an awful lot of gotchas that says that the science behind this and the Egyptian study are just garbage. And I'll let you go. Um,
Yeah, John, I think so. There's two main studies that you're kind of referring to. There's an Egyptian study where UK and Sydney doctors are saying the data is, let's call it manipulated, and
yep.
therefore, and manipulated in such a way it biased the results to show some promise for ivermectin. When reality is it wouldn't. They clearly state if you had just they had some redundancies of data, and if they just simply assessed the data the right way, it would have fallen out and would have shown that it truly has no benefit, which it doesn't. The second one, the Argentinian one, that one gets very funny because when asked about how he administered the drug, it was the honor system, John.
Yes,
That's the highlight I want to make on that one. When you hear like administrations of drugs on the honor system, I mean, look, I think that's okay, but actually it's not okay at all. That just means the study was not really done, John. That's kind of what this is. I'm not sure what the angle is on some of these things, other than notoriety and fame. I guess I do know the angle. That's the answer.
Sure.
But the problem with all this is you're now perpetuating this anti-vaccine movement, which I'm not against some of that. It's just if these anti-vaccine groups understood the validity of the data they were getting, they wouldn't be as supportive of it and as pounding of the desk as it's starting to gain momentum. And really, we need to stop it as soon as possible. I mean, John, I'm going to take a small breath here and kind of let you jump back in because there's more to these stories, but like that's my 2 cents on it.
Sure.
Both of these studies are things that are propagating and perpetuating this lie. If they were really, John, I'm sure there are some remedies out there that are helpful. These two are not, though. Let's be very clear. This one is not.
That's exactly it. I think that's the big danger here. We're obviously pro-vaccine. I'm totally on board with the vaccine, but also we want to know there are other therapies that can help. So we know that there's the Regeneron and Vir antibodies that help. The Lilly antibodies help when you're in a hospitalized setting. But
John,
you want therapeutics that can help downstream. If you, for example, you get infected before you're vaccinated, you want to know there are other medicines available. But ivermectin isn't one of them. But the danger is because the people read this and think, oh, I can go for that. They're going to take something that isn't effective, and they're just going to get sicker, and so it's not helping them at all.
Yeah, and John, we're working on 3 or 4 COVID therapeutics for late stage disease, right? I think that grade is like 1 to 8 or 10. They have some scaling, and we're actually doing clinical trials where, just as you described, people are making the investment. They're looking physician-driven, physician-led studies. None of the this ivermectin is physician-led at all, and those that are saying, "Oh, good Lord, this doesn't work at all."
Yes, and so let's actually jump back to the Argentinian study and talk through some of the flaws of that. So basically, I picked out some stuff. The number, gender, and ages of study of participants were inconsistent as you read through the paper. A hospital named in the paper as taking part in the experiment says it had no record of it happening. Health officials in the province of Buenos Aires have also said they have no record of the study receiving local approval. Then if you read more about it, the person who was cited as running the statistics for the study, she said, actually, I didn't run any statistics. I had nothing to do with it. At which point, Hector Carvalho, who was the lead on it, who is a retired endocrinologist and professor of internal medicine, they didn't actually do the statistics. So he can't even keep his story straight. Further down, there's no other talk of who provides the statistical data other than the statistician who we've already identified is not participating at all. It's just. I'm trying. It's just amazing.
It's mind blowing, John. Right? Yeah, it's mind blowing. This now you're not only is one of the co-authors on there saying, "Oh, I didn't do any stats on this," and that's the only one he lists, right? And then there are now 4 other hospitals that said, "Oh, we didn't participate in this study with this individual." John, it's just garbage. It's complete.
Yeah. So, and another thing I want to point out is that it was published in a journal. So not all journals are equal. So you can look online and you see what looks like an official journal. Some journals are more equal than others, to quote Animal Farm. But this one, it's been going, I think, since
2019.
2019, and it's published 10 articles in that time. That's not a serious journal. Plus, you have to pay to have your articles published. Again, that's not a serious journal.
Yeah.
So, it's...
John, you're talking about impact factors, and impact factors are important things. Not my favorite topic of the world, but it actually, in this setting, is exactly what you mean. But this journal, I imagine, has no impact factor.
Sure. Yeah. So, ultimately, but the trouble is that this is what is being cited as proof that ivermectin works, and it's one of those things that if you're coming across something out on the internet that contradicts the vast majority of what's been talked about in terms of COVID treatments, be careful with that
Yeah,
information,
John, it
because
should be in Science, right? Or Nature, or
exactly,
maybe
yes,
even
yes,
Experimental Medicine or
yes.
PNAS, but not this journal. Yes.
Yes, yes. I mean, when you're talking about ivermectin and the FDA says don't take it, trust the FDA rather than a retired endocrinologist in Argentina who won't share his data with his co-authors, won't share his data with other reviewers. He took the co-author off, who sort of said, "I'd like to see the data," and then backed out. If you've got a paper and you're taking co-authors off, it's still it's all these flags. Every single thing about this paper is full of flags that says this is nonsense. This shouldn't be discussed in the mainstream, but it's taken on a life of its own.
I mean, John, we can continue. It has taken on way too much of a life of its own, and even personally. But so, John, we talked
One
about those first two. Go ahead. I
Yeah,
was
just one last
just
thing. I mean, like you say, that this BuzzFeed article is great. I mean, what they talked about 4 hospitals carrying out the study. For one of them, no valid contact information could be found for the fourth hospital, Centro Medico Caseros, which appears to have no website or working phone. It's like, whoa, that adds real credibility.
yeah,
So,
John. It's the hospital de Dr. Warino here. It's in my basement.
yes. So anyway, yeah, the point we wanted to make here is, there's been a lot of talk about ivermectin, but ultimately, there isn't really in terms of the pro ivermectin um, effect, or the pro ivermectin therapy, I haven't found anything that's really credible. Though people will cite a study that has been done by the Institut Pasteur, who I would honestly view as a
Hmm.
hugely credible
Hmm.
institution. And I've got no fault with what the Institut Pasteur have done, but people are extrapolating from what the Institut Pasteur published. Basically, the Institut Pasteur in France did a study in hamsters, which is great. We know hamsters are excellent tox species in terms of building that initial data for potential COVID-19 efficacy. So that's fine, but this is on the Institute Pasteur website. They basically said the scientists observed that taking ivermectin was associated with reduced inflammation of the respiratory tract and alleviation of the related symptoms. The treatment is also associated with protection against loss of smell. However, the results did not show how ivermectin affects the viral replication of SARS-CoV-2. The findings of the study suggest that ivermectin could be considered as a potential therapeutic agent. So that's a reputable research place saying we've done a study in hamsters that says it merits further study. It certainly doesn't say go and start taking ivermectin because it looks like it might do something for hamsters. But I certainly have seen Facebook posts that have made that leap,
Yeah.
where people are pro ivermectin. They talk about Institut Pasteur because they know that's a credible name. They're not talking it's a hamster study. They're basically saying Institut Pasteur has said this is a drug that's got it's got legs, and it hasn't. And so finally, what we really want, the final study that I want to talk about is actually one that's done by supervised by McMaster University in Hamilton, Canada. And this study, I think, is similar to the way the Recover study was done in the UK. And the Recover study was the one where they had a lot of patients, where they actually showed that dexamethasone was efficacious, and they showed a lot of other agents weren't efficacious against COVID-19. So dexamethasone has been, obviously included in the treatment regimes for people. They did a study, placebo controlled, so it was a 50-50 split between placebo and treated. Ivermectin, it's 1,500 patients, so it's not huge, it's a decent size, but they said basically they saw no effect either of placebo or ivermectin in terms of treating COVID-19. And actually, I think the lead scientist, a guy called Mills, and I'm not sure what his
Yeah,
first name
Edward
is.
Mills. Edward
Edward
Mills.
Mills, yeah. Edward Mills said we actually started out on one dose, and because of pressure from other people, we actually upped the dose of ivermectin. And I saw a while back saw their sort of plan for different regimes they were checking, and they went to 3 times the dose of ivermectin and still found no effect. And so this data hasn't been published yet, but it was presented at an FDA or CDC conference.
Yeah,
So
John.
it's in a credible forum, you know.
This type of setup that they did is the one we all want to see.
Exactly.
And it just clearly proves that it doesn't work as a standalone therapeutic. It doesn't work as a co-administered therapeutic. It doesn't work in any way, shape, or form. It's just gobbledygook and craziness. People are grasping at straws. And even, I'm even going to quote Mills. He said, the advocacy groups have set themselves up to be able to critique any clinical trial. They've already determined that any valid, well-designed clinical trial was set up to fail. So they're pulling the conspiracy card, John, right? That's what these advocacy groups are saying. And it's unbelievable. Suddenly these suddenly people online are clinical trial experts. John, I feel like I've been studying clinical trials for 30 years and I'm still trying to figure them out, right? Like, oh my God.
Yes. I think that's it. It's suddenly, you see, and it isn't just on COVID, but I mean, obviously in watching the UK, you have the pro and anti-Brexit camps. But anytime any industry expert, and particularly as the UK is seeing sort of supply line issues at the moment, will say Brexit is a big deal. The anti-camp always said, "Well, I know much more than this industry person because obviously I have my opinion," and it completely counters that. And there's too much of that. It's like the voice of experts is somehow lost in the complete din of everyone's opinions, and it's
Grasping at straws,
yeah,
John. People
yeah.
they just want to hear, and I get it. There's people are crying freedom in the face of some of the mandates and so forth. But I mean, I just think we're all overlooking. Well, we're not going to talk too politically, but when you think of your freedom, that comes in many flavors and spices, so to speak. And getting a vaccine, which we've historically done. I mean, you can't get into a school system without getting a standard battery of vaccines. So we've all got them here in the U.S. I think there's just a lot of misperception, a lot of, I think, anxiety and stress. But John, this is the one and only time we're going to talk about this drug because it is not effective. And I even have a couple family members who are, I don't want to call them anti-vaccines. They just historically haven't taken the flu, and they just feel like they don't want to get it. Although I do think they're moving towards it. I feel like my brother can't get, he needs a little surgery, and I don't think he's going to be able to get it until he gets vaccinated. So he's actually scared.
Mm-hmm.
And so he brought this up to me, and that's when I think we talked a little bit offline. He was convinced by, a podcaster here in the U.S. who took it along with, I don't know, a biologic by Regeneron, and miraculously he got better. That's what got him better. I had to explain this to my brother that, if you put, I don't know, calamine lotion or poison ivy cream on your skin, along with taking a therapeutic from Regeneron, you're going to feel better, right? And it has nothing to do with calamine lotion, right? So
Sure, yeah.
It's completely fabricated. And I'm not sure where it comes from other than I think it just fuels the, you don't need the vaccine movement. And that's just crazy. It's crazy. And John, I'm going to stop. I don't want to talk. We should probably move on. We spent a good solid 15, 20 minutes talking about this, but I think it was important to bring it up. I got no closing parts other than don't ever take this drug. Okay. And whatever you do, my brother was even thinking about going to a vet because he lives out in Colorado, and he's like, oh, my buddy down the street has some of this and he's taking it. And I'm like, Rich, no, you got to stop. Do not take a pill. And that's why emergency room visits are off the charts. These are for 2,000, 5,000 pound animals, and you're popping in a pill the same dosage. Oh, craziness, John. I mean, it's really making me a little, it's troublesome.
Yeah, it's one of those things that take it if you've got head lice or whatever it's for. Don't take it to treat COVID-19.
It's almost as bad, John, sorry to talk over you. It's almost as bad as people who are buying pills online that have fentanyl in it. It's
Yeah.
almost as bad as that. It's not, but fentanyl's killing people, right? This drug is hospitalizing you. I don't think it's quite killing them, but it's crazy. It's just, I mean, suddenly you think the person on the other side of a internet thing or some sort of post or podcast knows more than the physician that's sitting in front of you. I don't know, John. It's just bizarre.
Yes. Yes. So we'll move on, shall we? Yes. So the next article, I think this could be quite brief. Actually, it's talking about 1 in 5,000. This was a New York Times, actually an opinion piece, but it's sort of talking about CDC talked in July that vaccinated people with Delta variant of the COVID virus carried roughly the same viral load in their noses and throats as unvaccinated people. So it seemed to suggest that even the vaccinated were vulnerable to getting infected and passing the virus to others. And, the media has been all over kind of things like this and talked about instances where this has happened. However, I should have written his name. Sorry about that. It seems that actually what has happened is that although you may have the viral load, infection from COVID is really still low in vaccinated people. They was talking about data from Utah, Virginia, and the King County in Washington State, which includes Seattle, saying that really it's about 1 in 5,000 vaccinated Americans have tested positive for COVID, and actually in states where the vaccination rate is high, that could be as that number could be as low as 1 in 10,000. So it's one of those things that it's talking about. Really, now the data is coming out, it's really illustrating that how the vaccine is doing how the vaccines are doing their job and are really effective.
Yeah, I think I feel like we talked about this a little bit in terms of just because you're vaccinated, that's not like you're not in a bubble. It's not a bulletproof vest per se. It is
Sure,
armor. It's
absolutely. Yep,
armoring you.
yep.
And then I think as you're really the Delta variant came in and it really worried us. What's fortunate about it is a lot of people, you can still have, I guess, some sort of positive test for the virus, but not actually be sick or propagating the virus, right? Because it's just everywhere, right? I don't want, again, we're breathing it in, it gets stuck in your nose canal. Whether you're vaccinated or unvaccinated, we all have like a certain amount of like probably viral particulates that are coming in and out of us. And then those that are unvaccinated, well, that leads to infection. Those that are vaccinated, it just means you might pop up positive on a PCR test, but you never really get sick. And that's what this study shows, John. Like the numbers are those that are actually coming up positive. And when I say sick, they don't get hospitalized, right? They may have a mild
Sure,
bladder or
exactly.
something
Yeah,
like that
yeah.
versus, so I'm just going to, the numbers read like 1 in 5,000 or 1 in 10,000, depending upon where the studies are. But if we just use an example of, if you took 5,000 people in King County, Washington, and that's where what part of the study was, and 5,000 unvaccinated, 5,000 vaccinated that tested positive, it's only like 1 in the 5,000 vaccinated is going to get sick, and then it's a much higher number in the
Yes, exactly.
unvaccinated.
Yes,
And
yes,
that's what this
yes.
is about. I think
It,
this goes back to like why you need to get
Yeah,
vaccinated.
yeah. I think it's key to it's really saying because of stuff being reported in the media, people may get alarmed about the potential risk from the vaccine that they're still getting infected, but ultimately those cases are really low. And yes, you're right. The unvaccinated risk is much higher. Like you say, the vaccines are doing their job.
Yeah, I mean, I think I hope I don't worry about things when it's everywhere. The reality is just keep. I hope people are reminded that social distancing is still important. Wash your hands when
Yep.
make sure you're cleaning the doorknobs. I mean, I hope your guard hasn't gone down. Some of the practices that were implemented by people, I always did, and my
Sure.
wife and son now they used to kind of think I was nuts. They're a little more inclined to understand why hey, maybe bleach your phone off periodically, or bleach wipe your keys and stuff like that if you're out. I still practice that, John. I still try to take my shoes off outside the house. Even just don't drag it around with you, right? Like you're not, the thought that you're never going to come in contact with it is, I don't know, that's craziness. But anyhow, John, I hope everybody still remember, and I mean, carry a mask. Even if you think you're susceptible, just put a mask on. When you see people on with a mask, John, doesn't that make you think, oh, I'm going to stay away from that person, right? So there's a lot of things you can do to keep yourself healthy if you're worried about this.
Sure. Absolutely. Yep. But most of all, you get yourself vaccinated, and it
Well,
definitely
that
takes the
helps.
worry. It reduces the worry. But
Yeah.
let's move on, because actually, there's some interesting scientific stuff emerging. I just threw so we talk about your research in the past. I threw this one in because it actually touches on some stuff I did, and a subject near to my heart. But it's actually the link between COVID-19 rattlesnake venom and a killer enzyme inspires treatment target. So this is all very early stuff. It's actually a University of Arizona professor Floyd Chilton has been studying an enzyme related to rattlesnake venom for 30 plus years. Now now he has evidence that the enzyme could drive severe forms of COVID-19 and provide new target for reducing mortality from the virus. Snake venoms are fascinating. I actually um, came at them from an analytical chemistry point of view, where I was working on my postdoc, and we had um, uh, separation technology that could, had the real power, really high power in terms of separation of mixtures, and combined with mass spec, you could learn a lot from them, and so I at the time bought a whole load of snake venoms from Sigma, and did analyses. And when you actually looked at like historical papers, I had one of the snake venoms was from the black mamba. Historical papers said there's about 28 peptides, which are about a 6,000 7,000 molecular weight in snake venom. But using capillary electrophoresis with mass spec, we could see about 100, at least 108. We couldn't see more than that because the mass spec, the mass range of the instrument was limited. So I'm sure there was more to look at. But from a academic point of view, trying to dig into what is in there and what it constitutes is fascinating. So that's my little snake venom piece.
Yeah.
But this is just very quickly. Floyd Chilton worked with researchers at Stony Brook and Wake Forest to look at blood samples from 281 COVID patients, and they discovered that levels of the enzyme secreted phospholipase A2-2A could predict which patients were likely to succumb to the disease, and that's how we're then looking at this could be a potential treatment target. So it's an early piece, but fascinating how again what looking at nature can actually
Yeah.
inspire how we move forward with medicine.
Yeah, this is how medicine's supposed to be researched,
Yes,
John.
yes, yes, exactly.
Like it makes sense to guys like you and I when you hear about venoms or even like poison ivy antigens. They're for the longest time they thought they could be anti-tumor effect,
Sure.
help with
Yep. Absolutely.
immuno-oncology, and they actually held some promise. I think the problem with it is um, all of us are going to respond differently, and they're just a little too potent. They're
Yes.
almost too
Yes.
potent
Yes.
naturally. So
Yes.
they when you get it's almost like a chemical reaction when you see something crash out and go from solid to from liquid into solid phase. That's they can't figure that out, and then as soon as you do it, oh, that's just for this one patient. What about the other 1,000 I want to do? It might be a different equation. I hope that made sense what I'm saying, because it's just I might need a microgram of a poisonous venom, and you might need 10 or something
Sure.
like that.
Yep.
So it gets
Yep.
very hard to study these things because they're just so potent, and everybody's immune system is so different. So
I think that's part of the point. Is like these things are such the venoms are such complex mixtures. There's a lot of different agents who all have different jobs. You got the blood
yeah.
thinners. You got obviously there's other toxins.
Well,
I mean,
that makes it that much harder, right? So
yeah,
when you
it's
get in there and they start treating people, it's hard
true.
to
Yeah.
discern what the heck
Yes.
happened too.
So how do you hone in on that one piece, that one peptide that actually could have a therapeutic effect? And obviously it's been done because you've got Byetta, which was based on the saliva from the Gila monster.
Mm-hmm.
It's an
Mm-hmm.
anti-diabetic drug. But
Mm-hmm.
this is where this is. This is similar. The enzyme identified is similar to an enzyme in rattlesnake venom that can bind to receptors in muscles and paralyze them.
Well,
Um,
this is so, John. It's actually a biomarker, right? This could be a
sure. Yeah,
potential
absolutely. Yeah,
biomarker.
yeah, absolutely.
That's
Yeah,
what it's really showing. This phospholipase A2 group 2A predicts if a patient will succumb to disease. So then that becomes a biomarker, which is great, right? Maybe there's a way to say, oh boy, you're in hey, you're in stage 3 of COVID, but this enzyme's high, you're headed towards stage 8,
Sure.
let's cut it off.
Exactly. Yes.
Then,
Yes. Exactly.
that's one incredibly, remember, information is very powerful in a clinical diagnostic setting or clinical diagnosis setting. Then the other piece is, can we capitalize upon it, and is it a therapeutic? So I mean, this is the story we want to read about.
Sure.
These are the
Yep.
ones we want to put on the podcast.
Yep.
This is exactly what science. When you hear, oh, it's all the answer to cure everything somewhere on the planet, this is an instance of it.
Sure. Absolutely. And that's why I thought it was a nice, interesting one to touch on. So we'll move on, and actually, it's a similar thing about the link between we're moving on to non-COVID topics here, and this one is actually unraveling the link between migraines and protection against diabetes. And actually, this I was not aware of this, but basically, scientists have long known that people with type 2 diabetes are unlikely to develop migraines, and migraine sufferers have a low chance of developing diabetes. So my wife is a migraine sufferer. So I thought this was fascinating that, as you get older, obviously you're looking at what is going to hit you, and the doctor's already having to go at me about possibly being pre-diabetic. I don't have migraines, but my wife does, so that's one thing she really doesn't have to worry about.
John, I'm going to interrupt you. We're in the same boat. My wife gets migraines, and I don't. I've never had one. I'm not pre-diabetic, surprisingly. So this is funny. You and I are in the same bed. I did not know this either, John. I did not know that type 2 diabetics are unlikely to develop migraines. And obviously, I did not know that migraine sufferers cannot be diabetic. But on the surface, it makes a lot. It's all through the negative feedback through your. It has a name for it. It escapes me the name, but there is a pathway that makes sense as to why this wouldn't occur in these disease states. But continue with the story.
So yeah, so it's basically scientists at University of Tennessee may actually have identified the link between those two very different disorders. I'm actually reading directly from the article when I say that, and it's two small proteins, CGRP and PACAP, that actually contribute to migraines, but also drive the production of insulin. And this was all done in mouse studies, and they found the link between the two. So it's so. Let's see. So CGRP lowered levels of mouse insulin to that potentially offsets insulin resistance. That's a hallmark of type 2 diabetes. And it also found that PACAP regulates insulin in mice based on glucose levels and promotes the growth of insulin-producing beta cells, help preventing existing ones from wearing out due to overuse. So so they can see what these peptides are doing. That actually those have already become targets for anti diabetic medications. So not anti-migraine medications. So there's actually already anti-CGRP medications already on the market, with anti-PACAP migraine medicines in development. But then the researcher who was talking about this says, well, my concern is if we disrupt that too much, then we may actually bump those people into the possibility of getting diabetes. So it's fascinating because it's identifying the pathway, but also be aware of the risks of treating that pathway, because it's everything in the body's interlinked, and it's fast. It's like I said, this, you're right. This is the stuff we want to discuss.
Yeah,
Because
and it's the duplicity of it all, right?
yes,
So
exactly.
it's a dual-edged sword, is how I think of it. But it's the hypothalamic pituitary adrenal HPA axis is what I was talking about, and it's all driven through ACTH, right?
Sure.
So this is, and it's right in these pathways. And so, John, what we're driving at, the take-home message is, a lot of these anti-migraine medications, and they're on the one. I think one just got approved by Amgen, or maybe
I think it's like Lilly Amgen.
a couple of them have.
Yeah,
and that's exciting. My wife was participated a little bit in some of that. She took an injection or something as a part of a clinical trial, and it really helped her, and she's very excited about it. But now I have to go upstairs and remind her, or not remind her, I have to go upstairs and inform her she might be susceptible to type 2 diabetes now. Which is just awful, because she would, I don't know about you, but my wife would trade getting migraines, maybe once a once a month or twice a month. She gets ones that are debilitating. She doesn't quite qualify for, kind of a chronic migraines, which is awful. But I wonder if she would switch that for type 2 diabetes. Maybe not.
Yeah,
But good
I
Lord,
mean,
John,
I think
that's sad that this happens. But this is what
the-
this is what science sounds like. Not go get a vet medicine from some quack
yeah,
doctor down in
yeah,
South
but
America.
we have to emphasize this is only a potential risk of these migraine medications. It's not known. It's just that because if we're disrupting a pathway that has been
Yeah.
identified as um, as agents that can help address diabetes, if we're knocking them down, we just need to be careful.
That's the
That's
way it should
exactly it.
work, right?
Yeah,
Like you should have
exactly. Yeah,
that forward
yeah,
thinking. That's
yeah.
what we want researchers to do. You want
Sure. Yep.
researchers that are
Yep.
saying, "Hey, I may be fixing this over here, but is there any collateral damage? Is there something
Sure.
happening over
Yes.
here?" And
Yes.
that's what we need.
Yes. Yes. So anyway, I yes, it's like I say, it's really interesting, particularly if you live with a migraine sufferer, and I mean,
Yeah, it's a
once
good story. I think it's one that it's very promising stuff, but there's the, hey, you know what they'll do? They'll find a way to
Mhm.
disrupt the pathway and not cause the diabetes. They'll engineer it the right way.
Sure,
They'll solve it.
exactly.
It'll
Yeah,
get
yeah.
solved.
Yes, because yeah, because what was said by the principal researchers, you can't inject CGRP and PACP into the body as therapeutic strategies for diabetes because these peptides actually cause migraine pain. So it's yeah. That, like you say, is the double-edged sword. It's like if we did this, we could help diabetes. Oh, now you got migraines. If we give you the anti for these, we'll help your migraines. But oops, what does that do to
Yeah,
potential
I mean,
diabetes?
nobody wants to go
But
from
it's
high blood sugar into chronic headaches, right? So I think we're
yes, absolutely not. No, like I say, having lived with a migraine sufferer and seeing what they go through, I never want anything like that.
but
So
it's
I'm
very promising. I think with a
yes,
take
yes,
home there is this is
yep.
how science gets solved. And John, we got one last news article because I think it's worth mentioning. It's kind of
I
cool
should,
about these
I should got
drones.
we got two, but
We got
one
two.
they, both can be really quick. Um, and because both are really different and interesting subjects. This is actually one. The headline is "Drones Outpace Ambulances by Airdropping Defibrillators to Heart Attack Patients," and it's funny. This my wife and I think as with most people's viewing habits, you binge watch things, and we do both have a fondness for like murder mysteries, particularly British murder mysteries, and so we recently completed Midsummer Murders, which is set in rural England, and it's like this rural area based around this small town. I can't remember what the town's name is. Coston. There we go. But the murder rate in that area is just outrageous compared to the rest of the UK. But actually, there was an episode where they were doing an experimental. Um, so they were doing like they were doing a trial delivering drugs throughout the village using drones, and someone adapted one of the drones to kill people, which is that's the background to
Yeah,
that. But
yeah, yeah.
moving on to the study in Sweden.
I won't get a drone from you, John.
Moving on to the study in Sweden, it's funny how it leaps from the pages of fiction into actual practical trial. And this trial in Sweden was looking at how was it possible to deliver defibrillators to heart attack victims via drone, because the big deal here is the quicker you can get those defibrillators to the patient, the more chance you have of saving them. And I don't know what it had in terms of instructions for the person with that patient to help them actually operate the defibrillator. But obviously, if you've got ambulances and you get caught in traffic, if that ambulance is sitting not able to get anywhere, you're losing time. Well, in this example, there were a number of it was the Karolinska University Hospital in Stockholm. There are a number of instances where the drone did get there faster than the ambulance, and this is a very early study. This is just showing potential. They said, there were some cases where they couldn't send the drones out because of weather conditions and things like that. They only had a limited flying distance. I think it was a 5 kilometer range, but it's all about potential, because you know that once you show the potential of something in terms of concept, someone's going to build on that. And again, this comes back to what science is all about. It's not always the first person to do something that solves the conundrum. It's someone who sees that idea and says, oh, if I do that, I can make it a little bit better. Someone else sees their idea and says, I can make that a bit better. And through that stepwise process, you end up with something that could actually be very, very helpful. So I love seeing articles like this, because it just shows how how diverse medical research is, and how all the different ways people are thinking how they can help people. And yeah, I'll pass it
It's
over to
great.
you.
No, so what they did is they sent out ambulances along with this drone, and you're right, it could go about 5 kilometers. And then a wench would lower the AED down to the ground. And then I think there's instructions in there how to use it, John. Those are pretty straightforward. They're on the wall in every gym here
Sure.
in
Yep.
the U.S. And I think to your point, this is real powerful stuff. I mean, I wouldn't. We can get a drone to fly a lot further than 5 kilometers. We can get it to fly in almost any weather. It's just still to be determined just how helpful this might be. But I would imagine it doesn't matter if there's traffic or not. A drone is going to get to somebody's house a lot faster than an ambulance would. And is it cost prohibitive and things like that? I don't know, but I like what Sweden's doing, and these are exactly the type of stories that we want to report on. This is what we
Yeah.
love.
So,
These are the things that it could lead to, instead of fleets for Amazon, right? And by the way, the reason why Amazon's not flying around with drones is people have already developed nets to take them down, right? That's why they're not out there flying around. You could steal that drone. I'd like to think no one's going to steal somebody's AED or even diabetic medicine. They talk about how it could be used in delivering of diabetes medicines, and you could see all along the way. I'm out in a canyon in Colorado. I get hit by
sure.
a snake. You talked about snake, venom. Hey, instead of having maybe that a drone could take it out to me. That's
Mhm.
great stuff. And John, I don't know if you knew this. I helped get the first drone into the country of Nepal to help bring fecal samples out of the bush into the laboratory, because they would drone them. What would be a couple days walk, they could take a drone back to the laboratory and get a fresh sample. Anyhow, I digress. But drones have been used in science for years now, is what I'm saying.
Sure. Yeah. So yeah, and so like we said, in 2/3 of the time, drones were there before the ambulance. So it isn't all the time, but certainly it's another tool.
Hey,
Particularly, you've
John,
got
if,
this.
if two
But
thirds of the time I can get 5 minutes faster to get
yeah,
my heart going again, I'm doing it, John.
yeah,
I'm doing
that's
it.
that's a huge difference to those. I mean, if we're talking
Yes.
12 patients, that's 8 of those patients. That's a huge
Yeah.
difference for those 8 people and their
The
families. So
people who get
yeah,
it immediately are gonna you have less long term damage. I mean, it's off the charts how
yeah.
beneficial this would be.
Yep. So last news topic
Certainly not the
is, I
least,
think,
though,
and
John. Not the least. This is a
this
very
is
good
this
one
is
too.
this this is finally our chance to put this subject to bed. Founders of Taiwanese biotech involved in stealing Genentech secrets plead guilty to conspiracy. We've talked about JHL Biotech before, and the lifting of documents where the Genentech name was scrubbed out and the JHL Biotech name was put on in its place, and they used the information to really accelerate biosimilars through their research line, and also to help try and sell the company to Sanofi for about $100 million. The two co-founders, Ratcho Jordanov and Rose Lynn, have actually now pled guilty to conspiracy to commit trade secret theft and wire fraud. So they each face up to a maximum of 5 years in prison, up to $250,000 in fines. So we talked about this in a little bit more depth in the past, but thought it was good to actually sort of say we can put a wrap on this one, because they've pled guilty to it. I'll let you chime in, John.
I don't have much to say other than they deserve what they get. I don't know if 5 years is
Hmm.
justified, but the people who commit these types of crimes really should go to jail for some period of time. I hope they see a jail for a small period. Just they're, as John, you and I are in an industry that integrity, honesty, and good ethics is paramount to our success. And These are the exactly the type of people who rose to a power of position and abused it. Just awful
Yeah,
story here, but in the end,
yep,
it all tends to find its way out. And I'm thankful that they're the rare people who do this type of stuff, or at least we hope. But it's just it's just straight up stealing, and it's no different than robbing a bank. They're bank robbers, in my opinion.
exactly. Yes, I've got no sympathy for them at all, and like I say, all they do is just affect the reputation of all of
Hmm.
us, and
Yeah.
yeah, it,
Causes people to want to
they,
take livestock medications instead of real medications.
yes, yes. The good thing is, I think in most cases these people do get caught, but I like I say, thought it was good to just wrap that one up and put it to
Yeah.
bed finally.
And,
So
and
yes,
and John, we can get on to our main topic here. We went really long in the news, but I think it was a jam packed news week and lots of stuff going on. So that was a lot of fun. But we want to jump right into it, or what do you think?
I think we probably should. Jeremy, do you want us to go ahead with doing the main topic? I mean, I don't know how long we're going to go, but
Yeah, I feel like we're ready,
Okay.
so let's just go ahead with the main portion then.
okay, that's fine. So yeah, main topic is we thought let's talk about one of the subjects that are near and dear to our heart, which is antibody-drug conjugates, the combination of targeted delivery potent treatment. And the reason why we talk about that is increasingly you'll see talk of antibody-drug conjugates or ADCs in the literature because you'll see more and more of these approved. Mostly they're mostly oncology drugs, so used in treatment of cancer. And the point of an antibody drug conjugate is to use the ability of the antibody to deliver a very toxic drug to the site of interest and take those tumor cells out. And I'm not sure when the first one was approved, but it's this is mainly research in the 2000s,
Yeah.
and it's now really sort of getting quite mature, and we're seeing a lot of candidates come through.
Yeah, John, it's fascinating that this dates back to almost 20 years ago when people started taking monoclonal antibodies and putting small molecules on them, hence the antibody drug conjugates. And why it started in 2000 and why are we seeing success in 2020 or 2021, or really in the last couple years? A couple things. The antibodies themselves have improved the technology tremendously, meaning 20 years ago, we could make a humanized antibody, but we can make it much better now, less immunogenic. We also make it with better binding, better affinity, better avidity. Sometimes the antibody can be binding too strong. That could be bad as well. So we've really, kind of engineered monoclonal antibodies. Therapeutics have made major advancements. And then on the small molecule side, same thing, right? So they've
Yes,
initially, when you dose someone with a small molecule, it's at a very low potency. But these ADCs are like 10, 100, 1,000 fold the potency of what is administered. So now they found ways to really kind of hone in on how potent that small molecule is. Also,
yeah.
not
Can I
only
just say
not only
something
that,
here?
but yeah, one last point is the technology
Sure.
to
Yeah,
the payload itself, the binding of the small molecule to the antibody that's exploded in the last 20 years. So this is how
yeah,
science works. 20, 25 years ago, people say, oh, why don't we combine these two very powerful therapeutics? Well, it takes time before they can suddenly hit the market. But John, these are curing people, as you know. I'll let you jump in,
yep.
but ADCs are a
Yeah.
major, major benefit for immuno-oncology.
Yeah, so yeah, so in terms of oncology, if you're taking an oral drug, it's going to be systemic. So although it's intended to treat a specific oncology in a certain region, it will affect other parts of your body. So you'll see side effects throughout. So it's
Yeah,
things
so
like
John, how
hair
that
loss,
works, right,
etc.,
is that
etc.
the tumor consumes a much higher level of nutrients. So it's going to take up that small molecule at a higher rate than the rest of your cells do. That's part of the principle around how small molecules work, right? That's
Sure.
you're taking
Yeah.
advantage of the tumor being super aggressive.
But like I say, because it's systemic, it will affect all your body. So that's why you're seeing side effects and why that can be tough for people to deal with. The attraction of ADCs is because you're going for a specific target. The We hope to minimize those effects to the rest of the body. It's really aiming to just attack the tumor and minimize the impact on other parts of the biological system. And like I say, this is the what we call the payload, the small molecule piece. These are so toxic that you couldn't actually administer these orally. They have. This is the only mechanism whereby you're going to be getting these into the body, but where they're proven to be effective against the um the the uh the cancer that you're dealing with. And also, part of what plays into this, the 20-year development time. You mentioned the small molecules, and a lot of those are they're fairly traditional names that, if you, as a analytical chemist, you see these names and you recognize them. But then you have, and they're changing, but also the linker chemistry between the antibody and the small molecule, that's developing with time as well, in terms of how does the linker cleave to help your, to basically help the small molecule break off and things like that. So there's a lot of research in terms of the antibody, the linker, and the small molecule to optimize that to then help treat the conditions we're talking about.
Yeah, I think Dana Fabo would be very proud of what's happened to some of his small molecules from back in the 50s and 60s, and how we've been able to make them into the toxicity levels that he wished he could dose a patient with, versus how we can put that on the framework of an antibody and deliver it to its target. I mean, it's not as easy as what I just said, but we're getting closer every day with being able to get that payload at an incredibly toxic
Sure. Yeah.
level to the destination and to the right place for it to do its action. And then John, maybe I'm stealing some of your thunder, but let's move on.
And actually, it's interesting. You were saying something I actually do want to build on. We we talk about these as a panacea. They're not. They haven't managed to eliminate side effects completely. They still have side effects, but like I say, a lot of it is they are getting very efficient agents into the body. It's interesting that one of the first ADC, Gentuzumab ozogamicin, which was under the trade name Mylotarg, was approved for leukemia in 2000, but was withdrawn from the market in 2010 due to adverse events, particularly hepatic side effects. But however, it was reintroduced to the market in 2017 with a lower dose and a new indication. So it tells you how people are, again, it's that what we talked about earlier. It's how researchers build on what's been done. So the early regimen was, they thought this dose is what's needed, but that has side effects. But further research says actually we can still get the effect with a lower dose and reduce the side effects. So it's all about learning your drug and what it can do for you, and yeah. So it's but so what I'm trying to say here. I think I'm just going off on a. I might have to.
No, it's exciting to you. That's all. It's just exciting.
Yeah. So not only are ADCs, we're looking. I mean, it's very much looking at cancer right now, but longer term, there's a hope that these will also be used for other diseases, such as inflammation-based diseases and just other medical therapeutic indications.
Yeah, John, I don't want to get too far ahead, but there are many things that fall into the ADC category, and not just like antibody drug conjugates. There are just therapeutic drug conjugates, and we won't talk about that too much. But there are ways to make the antibody use pieces of antibodies, for instance, and that has a lot of potential as well, because an antibody has two immunological properties to it: the FAB region and the FC. For those that understood what I said there. But John, the ADC field, you nicely started with how I think there was the approval in 2000, the taken off the market in 2010. I think it caused a lot of little, I remember going to an ADC conference almost 8 years ago, and the field was a little bit down at the time, I think in part because of some of what was happening here. But there were a lot of people and a lot of scientists, a lot of passion in that room. And I've been to a few others. The World ADC Conference is what it's called. been to that a couple times, and flash forward to today, and it's become highly successful. And a lot of it
Sure.
has to do with the design components. And John, I mean, you touched on them. Do we want to drill into what makes up an ADC a little
Yes,
bit more?
absolutely, yeah, yeah. So one thing I will say, in terms of where we've gone since 2000, we've got nearly 90 ADCs entered clinical trials in 2019. So that's what we're talking in terms of explosion in this area, how much focus there is on this, and why we'll be hearing more about them. But yes, let's move into the components. So they're basically, for the ADC, we've talked in passing, three critical components. There's the antibody, the payload, and then the linker. I'll let you talk about the antibody because obviously that's your piece.
Yeah, so the antibody itself has, as I mentioned just a few moments ago, two kind of components. The if you don't know, an antibody has it has two arms to it, and typically each of those arms actually binds to the same, let's call it, peptide region or sequence on a protein. Now, some of them, I'm going to throw a curveball at you. They call them bispecific ADCs, right? Now it can bind to two things, and you got a payload, very powerful stuff. But if we just stay along the ADC pathway, that will, and that's designed to bind to something on the tumor that's specific, some sort of epitope on the tumor that is not on normal cells, but just on the tumor cells. There's also, it can be done for hematological disorders. So a lot of times it targets something on a B cell that's gone crazy that isn't on other B cells. So it can help deliver the payload that way. So that's the magic of the ADC. It's a targeted bullet that should not bind to anything until it finds its resident ligand somewhere within the body. So now when you dose an ADC, instead of the chemotherapeutic just going into all the cells in the body, as John described earlier, it is going to stay on the back of that antibody. This is a pretty good sized protein where it's packaged to stay there and not be released
Sure.
until that antibody finds the tumor, right? And then what maybe people don't know is when the antibody binds to the tumor, it gets internalized. So now you should have hopefully not just a single antibody there, but hopefully we don't really know. Could be one, could be 100, could be 1,000. We were dosing millions and millions of antibodies into these people because it's a concentrated. When you get a MIG of something, there's 10 to the 23rd antibodies in there. And so those antibodies, not all of them are going to make it to their sites. A good portion of them will, kind of either be just distributed just through your urine and normal ADME type of processes. But what's happening now is they're getting specific enough and powerful enough to find their way to the tumors and bind. And so then there's a couple different mechanisms of action. One, they can get internalized. The other is they take advantage of the tumor microenvironment. And the tumor itself has think of it as like a thunderstorm going on in the body. It is not a nice area. It is a storm. It's a sea. You are like in the Bermuda Triangles if you're in a tumor microenvironment within your body. The tumors are doing all sorts of things to turn your immune system off, to repel your immune system. So if these antibodies make their way there, they can get internalized because it is consuming. We talked about that. Tumors are lovers of nutrients. They are consuming a 100-fold higher rate of uptake of fluids than your normal cells do. So they can get internalized. The other thing is some of them bind, and then because there's certain enzymes that tumor is secreting, they'll then get deposited, John. So there's two mechanisms of action. And that's where, so the antibodies themselves have advanced tremendously in the last, they came out, monoclonal antibodies were developed in the late 80s and 90s. It's on the cover of Time, I think, in like 1988 or something, as being the magic bullet to cure all. I think it was even earlier, maybe in the 70s. I feel like Nixon was president when that came out. But flash forward 30, 40 years of this, we've made great advances in so that your body doesn't find them to be foreign anymore. You don't make a lot of antibodies against a monoclonal antibody. That's an important advancement. And then, of course, the specificity and the ability of it to bind with a higher degree has become a major advancement. And so then there's the linking technology, John, and there's a whole slew of that. It's still evolving and so forth. That is an incredible industry in and of itself. They call them platforms, right? I'm a platform company. And then there's the small molecule. And I'll stop. I hope that was what you were looking for, John,
Sure.
but that's
No,
the premise of how it
it-
all happens. There's two ways that there's so much information. I hope people understand the power of an ADC. We're combining two major technologies, and then we're starting to get into some real heavy science when it comes to the mechanism of action of an ADC.
Yes. So yeah, I mean, just to touch on the payload of the small molecule piece, basically, it's the desire for that is to be potent enough to kill tumor cells at intracellular concentrations. And like I said, these are all toxic, and they either do that by causing irreversible DNA damage or interfering with the mechanism of cell division for the tumor cells. It's interesting that the article I pulled some of this from said generally there are 3 to 4 payloads on an antibody, so it's basically 3 or 4 of the small molecules on the antibody, and we call that the DAR, the drug antibody ratio. But actually, that's another piece where this is evolving. So we've been working on ADCs where that DAR has gone up, to around 8. I think we've been looking at. So again, it's the if you've got 8 on an antibody, that's 8. You can get eight of the small molecule to attack the cells onto with each antibody. If you're talking in the milligram, it's 10 to 20. Now that going from four payloads per antibody to 8, you're basically doubling the effect that you're
Yeah.
getting in to attack the cells.
Yeah, John, and I'll add the potency of the 8 can be a little bit less toxic because you can deliver more. Now, people go the other way. Some people are reducing it to 2, but that payload is almost covalently linked and it's much more powerful. So a
Sure.
lot of it is the journey, the sojourn, the journey to the site is where a lot of the technology, where the I guess the moment of truth comes out. Getting the antibody making its way to its destination through will inevitably probably happen. It's can it get there with the payload still on it, and how many of them are still there. So a lot of times, technologies that use 8, they have the linker isn't as tight. So well, not all 8 are going to make it there. If it does, John, that's part of what happens there. And if it falls off, it's not as toxic, right? But if you put 2 really powerful warheads on there,
Sure.
they better be on there very strong, and when they get to their site, they can be very powerful. And so what I also want to emphasize is when it gets released at that site, it will get uptaked, right? That's going to happen. So either way, there's a couple different mechanisms. The antibody binding to the tumor could trigger an immune response. That's
Sure.
one. The other is the payload itself. If it's still on the antibody, that's certainly going to, if it doesn't get internalized, it'll eventually get, as I mentioned, tumors are wicked. It will degrade everything. And then the tumor's going to uptake it. It wants to feed on what it's cannibalizing. That's the whole process. It's churning up any protein or cell that's near it, and then it's eating it. Yeah, they're
Sure.
nasty.
Yep. So that we touched on linker, and it is important that ultimately, because we want the linker to be designed, and it will be based on the small molecule that is the warhead. You want to, as much as possible, keep that molecule on the antibody to get it to the site of interest. So the linker chemistry is going to be a critical part of that. And I think what we should probably touch on, another reason why ADCs are so fascinating for us is as analytical chemists, it does, because it's 3 different regions of the molecule, and there's lots of different things going on. They're analytically challenging molecules to work with as well. For me as an LCMS person, you talk about the payload, but we want to know.
Yeah.
Measure free payload, which might be the payload that's released before getting the site, and you'll have an assay for that. Then you measure total payload, which is the amount of payload that did get to the site plus any free circulating prior to that. So that's 2 assays that we would immediately be saying we should be monitoring this, particularly in the early stage of development of a drug. And then on the large molecule piece, it'll be likely done by either LCMS or like LBA type approaches. You'll be looking at the antibody or the total ADC and to monitor what's going on with them as once administered to the body.
Yeah. No, John, this is drug development show, bioanalytical challenges. We're going to jump right in. You nailed it. There's for the small molecule itself, you need free payload is what we call that. Just, has it fallen off in the plasma serum, that measurements that we have. And then what we call a total payload. And that's a combination of what's free as well as bound. And when I say bound, was it on the drug? Is it on the linker? Is the linker drug out there in circulation? All of that becomes like a nice bioanalytical development. When I say nice, it takes an artist. I think of it as artistry, and we're very good at that, John. Right? We know how to make free and total payloads for ADCs. We do a large number of them, and then you very nicely touched on the other components. So there's typically at least 3 to 4 to 5 methods that are needed to do the pharmacokinetic and even the immunogenicity or ADA of an ADC. So we got the 2 LCMS methods for sure. They're pretty standard, usually done by LCMS, and that's the free and total payloads. Then you nailed it, John. There's the total antibody is almost always done, and that is typically done by LBA, but it can be done by biopharma mass spec or hybrid or affinity capture mass spec is what we call that. But it's typically an LBA method, and that's just what we call the naked antibody or the backbone antibody. So that's just the antibody without the payload, and we do measurements to look at total antibody. And a lot of people will do a combination of free payload, total antibody, and then they can figure out what their pharmacokinetics are. And even to some degree, you touched on it, the drug to antibody ratio, which is a very tricky analytical thing. But we don't necessarily have to look at that in the PK.
We don't have the technology for that. That's a high resolution mass spec thing.
The other one that is probably becoming more of the flavor of the agencies is the total ADC. And what we mean by that is the actual drug itself. That's the antibody with the payload. And there's a couple different ways you can do that. A lot of times we take advantage of having an antibody that can bind to the small molecule. Not an easy antibody to generate, but that allows us to look at total ADC by ligand binding or biopharma LCMS as well, which we do both of those type of method developments and validations at KCAS. So I'm going to repeat them again. You got 2 payload methods, a free and total. You got a total antibody and then an ADC. Total ADC is what we call that. And sometimes you do all 4, sometimes you do 3 of the 4, but somewhere in there you really got to make an investment in at least 3 methods. And then last but not least is the immunogenicity component, and that's the Ada assay. Now the Ada assay, you start as a total drug, and then over time you may look at different parts of the ADC, and that's done by inhibiting or some sort of epitope mapping of where the antibodies are being produced against the drug. So it starts as just the total drug, and then you can get into parts of, is there antibodies against the payload? Is there antibody against the linker? Is there antibody against the linker payload? So that becomes an onion. The ADC, the immunogenicity component of things becomes a much bigger endeavor than just a standard mAb. And of course, we don't typically do immunogenicity for small molecules, or at least not anti-drug antibody assays, because the body typically doesn't make antibodies against it. So I hope that gave everybody a little bit of a flavor there. I think there's a lot of things I said that might somewhat be confusing in terms of, how do we get anti-small molecule antibodies, for instance. Those become very powerful tools bioanalytically for us. So, John, I'm going to catch my breath and
I'm going to
let you get us back on track.
I'm conscious of time, but it's also interesting that if you're talking all these assays, and if you're doing them in one lab, there's additional challenges that this prompts. If you're talking multiple assays, there's an impact on logistics. Do you sample multiple tubes from patients to facilitate each assay? But if you don't, you've got multiple freeze thaws. What's the stability of each part of the molecule to freeze thaws? Things like we have to think about that. Do you then coordinate analysis between different teams? So if you've got one group doing the large molecule piece, other doing the small molecule piece? If you're looking at payloads, our challenge, the free assay is going to be looking at much lower concentrations than the total payload concentration. So that again is the demands for the instrumentation is different. It's what is the stability for all the components? There are a lot to be looked at this in terms of analytical complexity. It's taking your standard approach and upping it considerably because
Yeah.
there's all these other considerations you need to be thinking about.
And I'll give us a shameless plug here at KCAS. One of the reasons that separates us is we can do that under one roof. So we have a process to overcome those challenges.
Sure,
You talked
absolutely.
about them, John. They're very big issues. But if you've got your LCMS team, next door to the LBA team, they don't become issues in our lab. We can hand off same day. We can look at the freeze thaw. Hey, could it sit? All sorts of things that can be done. But you can exhaust sample, John. That becomes
Yes,
challenging in the ADC. When you've got the standard battery of things you've got to do, plus it's such a potent thing. I'm sure there's other blood chemistry work that might need to be done above it. We don't know that, right? But what we know analytically is it's like 5 times the effort of a normal biologic, for instance, or even a small molecule.
sure,
So you're already needing more volume, right? So all of that comes into play. You touched on the challenges bioanalytically, logistically, but then it becomes a HIPAA thing when you can't bleed 50 mils from somebody. You can only take like 15, right? So that becomes another problem. And if you got a lab in Wisconsin and a lab in Utah and a lab over here in, I don't know, Arizona, and you need 3 samples, that becomes a problem. And then, like you said, the timing and the stability of that sample. If you've only got 2 tubes, now I got to ship one. I got to ship all the. Oh, that becomes. It just. I'm not saying it can't be done successfully. That's not what I'm saying. It's just if it's me, I want to hedge my bets in the right direction. I want to mitigate that, and that's why you go to a lab that it's all in one shot.
sure, absolutely, yeah. So I would like to say we're obviously running long today because there's been a ton to talk about. Let's move on a little bit. Just talk about recent developments and approvals, and I've got a ton of notes, but I think we'll try and keep it brief. But actually, there's now 11 ADCs approved to date. Again, like mentioned earlier, focuses on oncology, and that still seems to be the bulk of where people are looking at ADCs at the moment. There's been two big developments recently. One was the approval of Trodelvy, which is sacituzumab govitecan HZIY, developed by Immunomedics, but now part of Gilead after a 21 billion acquisition. We talked about the acquisition because certainly at the time there was an awful lot. There was a lot of speculation. Had Gilead overpaid for this company, but this was approved in 2020 for metastatic triple negative breast cancer and metastatic urothelial cancer. And if you look through the ADC literature, a lot of the applications for these drugs is actually to try and treat cancers that have proved resistant to other therapeutic agents. And I remember going through it, looking at, I think, on whatever site, and for most of them it's like if you failed 3 or 4 different
Mm-hmm.
regimens, then the ADC comes into play. There's a lot of footage. The one to really watch is actually interesting is in HER2, which is trastuzumab deruxtecan, and this is developed by Daiichi Sankyo in Japan, and they're partnering with AstraZeneca, and this is seen as the blockbuster of the ADC world. I think they had an accelerated approval for it was breast cancer related. I'm not sure of the um, exact approval application, but they've got further studies going on. But there's a lot of excitement about this ADC, and actually a lot of the ADCs out there have, like we mentioned about the drug payloads that were fairly traditional in terms of payload molecules. We talk about, auristatins, SN38, which is the active metabolite of irinotecan. There's maytansinoids, calicamycin, sorry. But the Daiichi Sankyo payload is actually a proprietary derivative of exatecan. But it's sort of showing the excitement in this in this uh, in this field
Mm-hmm.
where it's going, and this is seen as the possible poster child. But there's a lot coming through.
Yeah, John, and I do know we're short of time, but I think one really critical thing, well, not one, a couple critical points to make. The approvals from both the Gilead drug and the Daiichi Sankyo drug, they don't traditionally, most of the ADCs had gone after a specific marker on a tumor like HER2 or CD38 on a B cell. They had gone after something that was, very targeted for a specific disorder. These two do not. They go after um, proteins that are expressed potentially on all tumors or any tumor type. So when you see it's treating triple negative breast cancer as well as urethral cancer, it's going to treat all sorts of cancer
Sure.
types.
Yep.
Gilead, I think, made one of the best purchases, even though initially we were like, oh my God, this is outrageous. But John, this drug is being distributed. It is so powerful that it was so beneficial during its phase 3 blinded trial that they immediately took all the people that were on the control arm and halfway through the trial stopped and said, "You're taking the drug." That's how powerful this drug had been to treat breast cancer, right? Or triple negative breast cancer, that is.
Sure. Yep.
Also, they're now expanding it into other drug types, and I'm sure Gilead would be more than welcome to tell you what their future is with this drug. I don't know it offhand, but there are tons. This can go after. They're probably struggling with, hey, where do we want to go?
Sure.
It's an
Yeah.
anti-TROP2, but there are some tumors that express TROP2 at a higher level, and what you're hearing in there is a lot of them have to do with your I'll call it the mucosal immune system from your mouth to your butt, right? And that's where urethral cancer is. And to some degree, breast cancer fuels into that thing, but they're really looking at anything, a lot those are unmet needs as well, John. A lot of those
Sure.
areas are very
Absolutely. Yes.
unmet
Yes.
because the mucosal immune system is so powerful that if a tumor can evade the immune system, it's just a hard area to treat. Solid tumors are very hard to treat, and that's what these things are going after. Both these drugs have targets that are against universally expressed tumor antigens, and that is where the real power lies, John.
Sure. Yep. Yep.
Not just
Yes.
for these tumor types, but I mean, John, I would say if you get if anybody's out there listening who's already gone through some chemotherapeutics, get on an ADC somewhere. I think this is the future. Or, we're not going to talk too much about CAR T cells. Those are my favorite, right? We love, oncology T cells. But John, any other? I feel like we've kind of hopped around. There's 11 drugs. We could go into this drug list a little more, but maybe let's wrap us up on some trends and some latest news. How's that sound?
Yeah. I mean, I think I think there's continue to be and continue to be research on optimizing
Yeah,
antibodies,
maybe
payloads,
improving
and
them. Yeah,
linkers.
that's a good point.
Yeah.
Go ahead. There's a lot going on with making them bigger, badder, and better, right? That's a huge investment right now in our field.
Exactly, and so in terms of talking about improving each part of the molecule, so like we talked about the Daiichi Sankyo, they have a proprietary small molecule that's moving beyond the um, more traditional payloads, but that other people were doing similar things. There's also focus on improving the antibodies, also looking at different and improved linkers. So this is a field that will continue to obviously it's moving quickly, and we'll continue to hear more about it. And I'm sure this will be a topic that we revisit in the not too distant future. But it's fascinating.
Yeah, no, and
It's
we do
amazing.
so much. We do a lot of it here, John. We could get Dawn Dewfield back on.
Sure.
She's doing a
Yeah.
lot,
Yeah.
or maybe even some of our LBA team, or maybe a combination of people, so we can talk more in depth on it. But ADCs, if you're not aware, they're the future of immuno-oncology. They already are. They're
Yes.
not going to be. They simply are. And with not there's all three of the components people are working on diligently. The monoclonal antibodies are getting better. The small molecules are getting better. And then that linker payload is really becoming a big industry, a big field. And what we mean by that is not only is it how you link the payload on there, There's ways of encapsulating it. There's ways of pegylating it. There's all sorts of things to help protect the small payload, protect the antibody because your body's very strong, help hone it in better. There's ways we're trying to look at ways of putting a dye on there and trying to traffic it the right way, right? I don't know if you know what I'm talking about, John. They can use some, and then they use a photo light so you can treat certain types of skin cancer this way,
Sure.
right?
Yep.
It's just incredibly powerful things that can be done. And we're scratching the surface on this. And I do think it should be a great
Exactly.
topic to re-up. But what I heard is 11 approvals, John. That's awesome.
Yes, exactly. And like I say, 11 approvals and 90 in clinical trials in 2019. So it tells you where we're going in the future.
I'm sure if you're going to see I bet you COVID delayed some of this. And I'm sure we're going to see a nice amount. We're going to track this, John, to see how many ADCs get approved in the next year or two. I bet you they're off the charts. They'll be that's probably one of the higher areas. The problem with ADCs is they can be toxic, so they got to rework them. They might take a little. I wonder what the timeline and cost is. I bet you it's longer than the standard drug.
Yeah, I'm not too sure about that.
I bet you someone has this information, John. It's
just
not you and I.
Absolutely.
But it would make total sense that the potential duration to get it approved. Now, you might get it fast-tracked and used in some conditional areas, but the approval of it, I bet you would be slightly longer than the normal, let's say NCE or BLA or even small molecule, right? And then the actual amount that it would take, I know bioanalytically it's a much larger investment, but good. I mean, as somebody who's aging, I don't care what the investment is. I want to see more of these. I want the money invested in ADCs. I hope in another 20 years, there's 50 of them, 100 of them approved, John.
Oh, sure.
We can personalize the ADC. Yeah. I want to see 5 types of the Gilead drug approved so that, not only is it, hey, we've got 5 iterations of, what's it called? Godolvi, right? Or Trodelvy. Trodelvy. We got 5 iterations of Trodelvy. Yeah, there's the Trop-2 with the payload on it, the SN38 payload. But hey, that's just for breast cancer. Oh, you have pancreatic cancer, you get this version of it. That's what I want to hear in another 20 years.
Yep. Yeah, I mean, it's interesting, like, people talk about we want a cure for cancer, but obviously there's so many different,
I say there is. There is a cure.
But yeah, that's exactly that's the point I was trying to make. I mean, to date, we're really talking about targeted stuff, but this is the potential to actually have, this is a potential direction to go where you can talk in terms of cure for cancer rather than cure for breast cancer, pancreatic, whatever. It's that making it a potential for making it a much more generalized cure.
John, if you had Hodgkin's or non-Hodgkin's lymphoma 30 years ago, it was a death sentence. Today, it's not.
Sure.
When people say, how come there's no cure for cancer? I assure you and there's still plenty of work to do in the leukemia and lymphoma state. Just like there's still lots of work to do in breast cancer, but lots of people are surviving cancer at a higher rate than ever. Now, Alzheimer's and neurological disorders, John, that's a whole nother podcast, isn't it?
Oh, exactly. Yes, exactly. Yes.
Well, this has been great, John. We're going to skip any news articles we might have. I'm going to jump. I just want to give you to everybody out there. I want to say congratulations, right, John? Did you finally get to see your son get married? I know it's been a long time coming. You can share that if you don't mind sharing that. I hope I'm not putting you on the spot, but everybody here at KCS is so excited you finally had that happen. I hope it all went well. I haven't talked to you about it. How did it go?
So, well, the background to this is obviously we were expecting a destination wedding in Jamaica in November, and both Thomas and his fiancée Amanda, now wife, they were getting a little bit concerned in August whether that would go ahead in November, because obviously, if they're asking people to go out to Jamaica, they felt some responsibility for people in terms of, exposure to COVID, etc. There was a couple of things happened. Jamaica announced that they were going into lockdown on weekends, and also I think the USA advised people it's probably not worth traveling to Jamaica at the moment, combined with hearing that the immunization rate in Jamaica was quite low. So they said, we're not going to do that piece. So they actually they did, ahead of the Jamaica wedding, they had a civil ceremony scheduled for September 16th. So that became the wedding day. It basically expanded everything, but it also meant we had to plan a wedding in 3 weeks. So there was a lot of scrambling, and huge kudos to my wife, who did a lot of running around working with Amanda to help coordinate where a reception, flowers,
Gotta
the
eat,
cake.
John. You gotta
cake.
eat something.
Yes,
Cake,
exactly.
dresses,
So,
photos.
so
There's a lot.
yes. So Amanda had actually linked up with a photographer who helped with the ceremony and said, "Well, in Boston, you can pretty much turn up at any public space, and you can have a wedding there." I mean, my son's wife is Jewish. He is, we're sort of very lapsed Catholic, to be quite honest, and so they did it in I'm trying to think, it's a rose garden near Fenway Park. It was a little bit sort of a little bit more freeform than I could necessarily deal with, but it was a really nice afternoon. Apart from this, we arrived at the site, and as we're driving down from Newton to the site in a taxi, you could see the rain coming down, and we arrived at the Rose Garden in this like fairly heavy drizzle. So my wife, and it's Kelleher Rose Garden. Luckily, it's next door to the Museum of Fine Arts in Boston. So my wife and I ran over to the shop at the art museum, and we bought a whole fistful of umbrellas, which were like with Monet and Van Gogh pictures on the umbrellas. And the women in the shop were like, "What are you doing?" I was like, "It's a wedding. We need umbrellas." So the shop staff were fantastic in terms of getting it all unpackaged and just being really nice. And so we shot at the museum back to the wedding. The wedding itself was really nice, and then there was a bit of a slight break. And when we did, the reception was a pub called Bostonia, and we found that sort of fairly last minute. We got the room to ourselves upstairs, and we had an absolute blast. So it was a really, really, really nice day. Just waiting for the official photos and stuff to come through. Food at the Bostonia was excellent. It's like my wife has been binge watching Four Weddings on Discovery over the last few weeks.
Is it four weddings and a funeral, or is that different?
No, it's four weddings where four brides go to each other's weddings and rate it, and then the highest rated wedding, they and their husband go off on a paid for honeymoon. And it's like, generally I look at this and the food looks terrible. So it's this big fear, like booking this place at short notice. What's the food going to be like? It seriously was great. And so huge kudos to the Bostonia, because that really helped make the day too. Because it was like a sit down meal, but still relatively informal. So people were like drifting about between courses and things. But yeah, just a really, really, really nice day. And both Thomas and Amanda are really happy with how it all turned out. They actually, the next day, Friday, they flew to Philly because they were in Philadelphia to watch yesterday's Eagles game, and we flew back to New York on Friday too. But yeah, the family are all very, very happy with how it went.
Now that sounds like quite an adventure, John, and congratulations. And it's not Jamaica, but Boston's a fine place. The Fenway area that you're describing, I think I know the Rose Garden you're talking about. Certainly know the Museum of Fine Arts. Sounds like it worked out just nice, and all's well that ends well, I suppose. But
Yep.
welcome to 2021, right? We're getting married.
Yeah, and
not as easy as it used to be.
And now we all start again planning my daughter's wedding, whenever that's going to be. So it's we'll learn. It's always interesting to go through it. You learn things to do and things not to do, and maybe I wouldn't do this, and I will definitely do that. But that's next on the calendar.
Yeah, my son turned 16 yesterday, so I'm a little ways away from marriage, or I hope a good ways away. But
you hope,
16 is one of those ages where I asked him, I said, "You feel any different today?" He said, "No." I go, "Well, you can drive your car now without other than school."
Sure.
So he's pretty excited about that. So, but no,
well,
John, this has been a great podcast, John. Yeah, sorry, go ahead. It's been fun.
I just said congratulations and happy birthday to him. So yes.
And then I haven't talked about how I did use my Uni pizza oven, which was fun. Oh God, that thing's great, John. My little pizza. I got to mention pizza to some degree. I can't let the fan base down. But this, if you're not familiar with an Uni pizza oven, it is a portable pizza oven that makes like basically brick oven pizza.
Sure.
It uses wood pellets,
Yep.
right? I know you're
Yep.
familiar with it, but John, I finally got the right tools and everything, and I whipped up 3 or 4 of the best pizzas we'd had, and I'm very excited to use it in the future. I can't wait to. Still got to work on getting that thing. There's some artwork to spinning it in there, and I'm getting better at it with each pizza, but I can't wait until they come out. I'll be printing them soon here, John, and they're tasty. Anyhow, John,
One other thing I'm going to say about the wedding. So, we actually had the signature cocktail for people to try, and the background to that is a long time ago. My wife got a recipe book by Nigella Lawson, and they listed. I think it might be a Christmas recipe book. Anyway, it lists this cocktail called a poinsettia, and it's Cointreau, champagne and cranberry juice, and so that's become a Christmas tradition in our house that we have it every year. It sort of helps kick off the Christmas day. My wife actually contacted Nigella via Twitter to say, have you got any suggestions for a cocktail? What would you suggest for a wedding reception? I think she came back with a Moscow Mule, which my wife was, that we like them, but now let's go with the poinsettia. So it still became a Nigella cocktail, but that was the signature cocktail for the reception, and I had plenty of those. They were great. So really,
those sound dangerous, John. Those types of drinks are very dangerous.
Yes.
They go down a little too easy, and after the third or fourth one, you just start thinking, "Oh, I can be okay," and then the rest
very drinkable. Yeah. Yes.
No, well, that's great, John. Congrats, and been a great podcast here, and looking forward to the next one. Which, speaking of which, let's keep it a mystery for everybody.
Sure,
absolutely. Tell everybody we got something exciting for our next podcast. Maybe a visitor or something, right?
Let's wind up.
Yeah.
And yes. Okay. Well, thank you very much. You're right. It was a great podcast. Looking forward to the next one.